Welcome to BiG Science Academy. I am Bikram Ghosh. So, let's talk about photosynthesis. We will continue from the first part. From the first part of photosynthesis, we have discussed about the basics of photosynthesis. Whoever missed the video, the link is given in the description below. So, let's start about the photosynthesis in this part 2 video. In this part 2 video, we will talk about the two phases of the photosynthesis. That is the light dependent phase and the second one is the light independent phase. So, let's continue. So, there are two phases of photosynthesis. The first one is the light dependent phase or the light reaction, and the second one is the light independent phase or dark reaction or the C3 cycle, and it is also known as the Kelvin cycle. So, let's see what is the light dependent phase or the light reaction. The light dependent phase or light reaction occurs in the thylakoid membrane of the chloroplast. As the name describes, it is the phase of photosynthesis that needs light. That means light is very essential for the proper functioning of this light dependent phase or light reaction. Without the light, this reaction will not occur. In this phase, the chlorophyll molecule gets excited by the photons of sunlight. The excited chlorophyll molecule then performs two processes. The first one is the photolysis of water, that is the light dependent breakdown of water, by which the water molecule generates 4 electrons, 4 protons and 1 oxygen molecule. The second step is the electron transfer by various carriers through the thylakoid membrane or along the thylakoid membrane, which ultimately generates NADPH and ATP. So, the end product of light reaction are ATP, NADPH and oxygen molecule. So, let's see what is the light independent phase or dark reaction or C3 cycle or the Kelvin cycle. The same reaction is named differently based on different scientific researches. So, the dark reaction or the C3 cycle occurs in the stroma of the chloroplast, that is the matrix region of the chloroplast. As the name describes, that this phase of photosynthesis does not require light, but this does not mean that the phase occurs in dark. Actually, the dark reaction or the light independent phase is the phase in which there is no direct involvement of light, but it needs the product of light reaction or light dependent phase to activate the dark reaction. So, in absence of light, the dark reaction will not occur, because in absence of light, the dark reaction will not get the light reactions products such as the NADPH. So, it can occur anytime in presence of light reaction products. If the light reaction products are given during the night time or in absence of light, the dark reaction will occur. So, it depends upon the presence of light reaction products and CO2. So, in C3 cycle, CO2 from the atmosphere and NADPH from light reaction is utilized to produce glucose, the photosynthetic product. In C3 cycle, the most abundant enzyme in the world, that is RUBISCO, is utilized to fix CO2 during the carboxylation step. So, here we can see in the picture that the stroma region is a site of dark reaction and that the thylakoid region is a site of light reaction. So, let's start with the light dependent phase or light reaction. The light dependent phase of photosynthesis is actually the phase where light is needed. as we have seen that in the dark reaction there is no need of light for the process. Second, light is needed to activate the chlorophyll molecules. Actually, the photons of light activate the chlorophyll molecules for their further process. Light is needed for the photolysis of water, that is light dependent breakdown of water to release electrons, protons and oxygen. The electrons are then carried by various electron carriers along the thylakoid membrane for the generation of two molecules of NADPH2 with the help of an enzyme we will see later in the dark reaction. These two molecules of NADPH2 is then utilized in the next phase of photosynthesis, that is in dark reaction. We will later see in this video. So keep watching. So this is the Z scheme or non-cyclic phosphorylation of the photosynthesis, or it is the light reaction phase of the photosynthesis. Here we can see that the two boxes, green boxes, one is photosystem 2 and the other one is photosystem 1 is present. Both these photosystem are dependent upon light for their activation. So here we can see that light rays fall upon them and then only they are activated. So that's why this phase is a light dependent phase, that is the light reaction of the photosynthesis. Here in the photosystem 2 we have seen that the light is falling, and in presence of light the photosystem 2 splits water molecule into electron, proton and oxygen molecule, which is known as photolysis of water. The electron is then carried to the photosystem by tyrosine, and the excited photosystem 2 then donates the electron to the first electron acceptor, which is the pheophytin. We can see here in the blue blocks, that is pheophytin. The pheophytin then gives the electron to plastoquinone A, which then gives the electron to plastoquinone B, and in this way these electron carriers carry forward the electron. Then from plastoquinone B the electron is given to the cytochrome B6F, and the electron is then given to the plastocyanin. Then the plastocyanin, which is actually a protein present on the thylakoid membrane on the external side or on the lumen side of the thylakoid, gives the proton to the photosystem 1, that is P700. With the help of light and the electron, the P700 gets excited, and the P700 then gives the or donates the electron to A0, a chlorophyll molecule, which is also an electron carrier. The chlorophyll molecule then gives the electron to the quinone molecule, which is A1. The quinone molecule then donates the electron to various iron sulfur clusters, that is here it is given the 3 iron sulfur clusters, that is FeSX, then FeSA, then FeSB, and the iron sulfur clusters then ultimately donates the electron to ferredoxin. The ferredoxin, with the help of electron and FNR, which is the ferredoxin NADP reductase enzyme, reduces 2 NADP plus molecules into 2 NADPH2 molecules, and the protons which are used in this reduction process comes from the photolysis of water. Here it is given by an arrow. Okay, so we now see that there are two boxes. One is the DCMU and the other is the paraquat. The DCMU or paraquat are the inhibitors of this non-cyclic phosphorylation process. DCMU inhibits the electron transfer between the two plastoquinones, and it halts the total phosphorylation or total non-cyclic phosphorylation process. DCMU blocks the photosystem 2 process or the photosystem 2's electron carry. The full form of DCMU is dichlorophenyl dimethylurea. Next, the paraquat blocks the electron transfer from ferredoxin to ferredoxin NADP reductase enzyme. Thus, it blocks the electron transfer in photosystem 1. So, this is the total Z scheme or non-cyclic phosphorylation, that is non-cyclic photophosphorylation. Here it is a picture of the book from Tej and Zeiger. We can see that the membrane bound protein complexes are present which play a key role in the cyclic photophosphorylation. Here it is, the picture describes the transfer of electrons and protons in the thylakoid membrane and generation of NADPH by reduction of ATP by phosphorylation, which is photophosphorylation. Here we can see that the electrons generated by photolysis of water at PS1 are transported through the thylakoid membranes by various electron carriers to ferredoxin to reduce NADP plus to NADPH on the stroma side with the help of the enzyme FNR or ferredoxin NADP reductase. Next, we can see that the protons generated in the lumen side of the chloroplast passes through the ATP synthase protein complex in the stroma side, to the stroma side, generating a proton motive force which helps in the production of ATP from ADP plus Pi. This is actually the photophosphorylation step where the phosphorylation is occurring, that is the ADP plus Pi is combined to form the ATP or the energy rich molecule. Next we will discuss about the cyclic photophosphorylation or the cyclic phosphorylation process. Here, as we have seen in the earlier step, that is the non-cyclic photophosphorylation, there are two photosystems involved. One is the photosystem 2 P680 and the other one is photosystem 1 P700. But in case of cyclic photophosphorylation, only one photosystem is present, that is the photosystem 1 or P700. The P700, upon excited by light, it gets to an excited state and donates electron. The electron is then accepted by the chlorophyll molecule, the A0. The chlorophyll molecule in turn donates the electron to the quinine molecule. Then the quinine molecule donates the electron to the iron sulfur clusters, that is the FeSX, FeSA, FeSB. The iron sulfur clusters then donates the electron to the ferredoxin and the ferredoxin gives it to the plastoquinone B. The plastoquinone B then gives the electron to the cytochrome B6F, which then gives the electron to the plastocyanin. And the plastocyanin, as earlier in the non-cyclic photophosphorylation, here also gives the electron to the photosystem 1. And during this cyclic phosphorylation process, ATP are generated. ATP molecules are generated during this electron transfer process. So, the cyclic phosphorylation is performed by plants to meet the high demand of ATP, which cannot be met by the non-cyclic photophosphorylation. So, to meet the high demand of ATP, plants often perform the cyclic photophosphorylation to generate ATP from the cytochrome B6F complex. So, let's see the differences between the two photophosphorylation processes. First, here on the left side it is cyclic photophosphorylation and on the right side it is non-cyclic photophosphorylation. First, only the PS1 or P700 is involved in the process of cyclic photophosphorylation, and both the PS1 and PS2 are involved in the non-cyclic photophosphorylation. In the cyclic photophosphorylation, photolysis of water does not occur. we have seen that there is no photolysis of water in the cyclic photophosphorylation. But in the non-cyclic photophosphorylation, photolysis of water occurs, and it is very essential we know that, because this photolysis of water generates the electron which is then carried forward. So hence there is no photolysis of water, oxygen is also not generated, but in case of non-cyclic phosphorylation, oxygen is generated. The source of electron in case of cyclic photophosphorylation is photosystem 1, and in case of non-cyclic photophosphorylation it is the water molecule which is broken down during photolysis of water. The cyclic photophosphorylation is inhibited by paraquat, and the non-cyclic one is inhibited by DCMU, as we have seen earlier. In case of cyclic phosphorylation or photophosphorylation, only ATP is generated, but in case of non-cyclic photophosphorylation, both ATP and NADPH are generated. So, the light phase or the light reaction has ended. Now we will discuss about the light independent phase or dark reaction or C3 cycle or the Kelvin cycle. It is very easy, so let's concentrate and let's see. First, the molecule ribulose 1,5-bisphosphate, which is a 5 carbon containing carbohydrate present in a number of 3 molecules, is converted to 3-PGA or 3-phosphoglyceric acid, which is a 3 carbon containing compound which is produced in 6 molecules number. This process is carried out by the enzyme RUBISCO, which is the ribulose 1,5-bisphosphate carboxylase oxygenase, and this RUBISCO enzyme fixes the atmospheric CO2 molecule into the carbohydrate molecules, and this step is known as carboxylation. In the next step, which is the reduction phase, we will see that the 3-PGA molecules, that the 6 3-PGA molecules is converted into 6 molecules of 1,3-bisphosphoglyceric acid. Here 6 ATP molecule is utilized to convert the single phosphate containing molecules into 2 phosphate or double phosphate containing bisphosphoglyceric acid molecules. The 1,3-bisphosphoglyceric acid molecule is then converted into 6 molecules of glyceraldehyde 3-phosphate, which is also a 3 carbon containing carbohydrate. This G3P is formed by the utilization of 6 molecules of NADPH2 and the excretion of 6 inorganic phosphate. So at the end 6 molecules of G3P is produced, but from here 5 molecules will enter into the C3 cycle's regeneration phase, and 1 molecule of G3P will go to produce carbohydrates or photosynthetic products which are starch sucrose, and these molecules are utilized in glycolysis for the generation of ATP by respiration. So in the regeneration phase, we see that 5 molecules of G3P are utilized to produce 3 molecules of ribose 5-phosphate, and these 5 carbon containing sugars are then converted into ribulose 5-phosphate, which is also a 5 carbon containing sugar. The ribulose 5-phosphate molecules are then converted into ribulose 1,5-bisphosphate by the utilization of 3 ATP molecules. So at the end of C3 cycle, we see that the ribulose 1,5-bisphosphate is broken down or converted into various other molecules, which are then again utilized to regenerate the ribulose 1,5-bisphosphate molecule for the next cycle. So in one cycle of C3 cycle, there is a utilization of 9 molecules of ATP and 6 molecules of NADPH2. But in actually 2 cycles of C3 cycle are performed, so in total 18 molecules of ATP and 12 molecules of NADPH2 are utilized. The first stable compound is 3-PGA or 3-phosphoglycerate, which is a 3-carbon compound, and in this C3 cycle RUBISCO shows carboxylase activity. The location of the C3 cycle is the stroma of chloroplast. So let's talk about the most abundant enzyme or protein on the planet Earth, which is the RUBISCO. RUBISCO is present in every plant's body because every plant needs to perform photosynthesis, and RUBISCO is the most important enzyme in the whole world because due to the presence of RUBISCO, the photosynthesis is being carried out, and due to the process of photosynthesis, the total living world is getting their food. So the full form of RUBISCO is ribulose 1,5-bisphosphate carboxylase oxygenase. RUBISCO has a dual property or dual functional property, that is, it can act as both carboxylase and oxygenase in different cycles of the plant. In the C3 cycle, it acts as carboxylase where it fixes CO2 molecule, but in C2 cycle, it acts as oxygenase where it fixes O2 molecules. RUBISCO enzyme has only one site to bind the CO2 and O2. So it can bind to either CO2 or O2 at a single point of time. This depends upon the environmental conditions around the C3 plant. When it binds with CO2 in high CO2 concentration, it performs carboxylase activity in the C3 cycle. But when it binds with O2 in low CO2 or high O2 concentration, it performs oxygenase activity in the C2 cycle. Okay. So here is the end of the photosynthesis part 2 video. Photosynthesis part 3 video will be coming soon. Thank you. Subscribe to the channel BiG Science Academy. Click the bell icon for the notifications of the next upcoming videos. Follow us on Facebook. The link is given in the description below.